Okay, so today I'm going to talk about a recent paper that was published in my lab in the Journal of Biological Chemistry entitled Age and Insulin-like Growth Factor Impact PCNA Mono-ubiquitination and UVB-irradiated Human Skin. This is, again, a recent paper from the lab, involved a collaboration with Dr. Travers, a dermatologist in our department, a medical student who helped collect some skin biopsies from people, and then a couple people in my lab, Rebecca Hutcherson, who was at the time a technician, and Amber Castellanos, a master's student. And so the purpose of this work was to understand why older people have higher rates of skin cancer. And one of our interests in the lab is understanding how cells and how keratinocytes in the skin respond to UVB radiation, and how that induces DNA damage, and how that may be different in older individuals. We know from previous research that there's evidence that the rate of removal of UV photoproducts from geriatric skin is slower than in young adults. But we also know that there's other aspects of the DNA damage response that may be different in older versus younger skin. So the purpose of this paper is to actually look at one of those other pathways, and that's PCNA mono-ubiquitination, which is a post-translational modification to this protein known as PCNA. It stands for proliferating cell nuclear antigen. It physically forms a clamp on DNA that helps DNA polymerase move along DNA during DNA replication. But it's known that it can be modified through this mono-ubiquitination, which is essentially an 8 kilodalton shift. It's essentially, again, a post-translational modification to the protein. And we know from the last 20 to 25 years of research that that modification is associated with recruitment of specialized DNA polymerases, known as TLS polymerases or translucent synthesis polymerases, that can be recruited to sites of DNA damage and help cells replicate UV photoproducts or other forms of damage. And the canonical DNA polymerases that replicate chromosomal DNA during S phase, including both delta and epsilon, they're unable to replicate across bulky lesions like UV photoproducts. And so these polymerases can be recruited, but they require a signal. And this modification to PCNA is thought to be involved in that. And although this is widely detected in the field, in vitro, in cell culture, from human cells down to yeast, it's never been detected in human skin. So the purpose of this paper is to actually see can we detect this modification in human skin after UV exposure. And so what we did was to take abdominoplasty skin discarded during routine surgeries, and then to expose them to increasing doses of UV radiation. So this was work done by Becca in the lab. So she took human skin exposed to increasing doses of UV, 0 up to 1200 joules per square meter, and then harvested, take small punch biopsies, about 6 millimeter in diameter, freeze the skin, and then when we were ready to analyze it, we harvest the skin, scrape the epidermis off, and do Western blotting for PCNA. And again, typically, we can detect PCNA mono-ubiquitination as a shift on an STS gel using an antibody against PCNA. And as we can see here, there might be a small level of some other form of protein up here, but it's very weak, and this is much weaker than what we would see in cell cultures. What we did was to use a new antibody that's available through cell signaling, which recognizes a specific modification on lysine 164 PCNA. In this case, we see with this antibody a nice dose-dependent increase in this signal. So we can clearly see PCNA mono-ubiquitination in a UV dose-dependent manner. We compared these responses in 3 different skin samples from 3 different individuals. And B here, this is looking at simply the ratio of PCNA ubiquitination to total PCNA, just to control for any differences in total PCNA. Since what can happen is, again, if the protein becomes modified, it shifts on the gel, and so you see less of potentially less of the unmodified protein. So we want to compare that ratio of ubiquitinated to non-ubiquitinated protein. In all 3 skin samples here, we see a nice dose-dependent increase in response, although there is significant variability. For example, sample 3 here, there's a much higher level of basal level of PCNA mono-ubiquitination, and then with UV it goes up. So to compare this, the responses between different individuals, we set the 1200 joules per square meter as a maximum signal, set it to an arbitrary value of 100. And you can see in all 3 cases, again, we see a nice dose-dependent increase. And using nonlinear regression, we calculated the dose of UVB that was required to induce half maximal response. And it varied from 525 to about 700 joules per square meter. So although there's again some variation in absolute levels of mono-ubiquitination, we can see in B. In C, looks like to be a pretty similar range in terms of a dose that's required to generate a half maximum response. So with this information in mind, we then performed additional experiments. And again, we were curious if we could see if there's a difference in this signal in young individuals versus geriatric individuals. And maybe that's why they may contribute to increased mutagenesis that will occur in geriatric skin. And so what we did was, again, collaborate with Dr. Travers, where we recruited 12 young adults, 12 geriatric individuals who came into the clinic, and we obtained skin punch biopsies from their skin, from their buttock, brought it back to the lab, and then exposed to UV radiation. In this case, we used 700 joules per square meter UVB. And then 2 hours later, 2.5 hours later, we snap froze the biopsy. And then what we did was then harvest the tissue, isolate the epidermal protein, and then do Western blotting for both PCNA mono-ubiquitination and total PCNA. And what we see then is in both groups of people, both in the young adults and the geriatric individuals, we see clearly a strong UV dose-dependent increase in this signal. So in both cases, it goes up with UV. But what we noticed was that the level of increase on average was a little bit higher in the geriatric skin, and it's about 1.7-fold or 1.75-fold or so increase. This is the average here in the young adults, and then in the geriatric, again, it's a little bit higher, suggesting that this pathway, this translation synthesis pathway, which recruits and potentially mutagenic polymerases to DNA to allow replication across UV photoproducts, that response is elevated in geriatric skin. So we wanted to better understand the mechanism by which this takes place. Previous research from Dr. Travers' group and my lab has shown that, and others, has shown that IGF-1 or insulin-like growth factor seems to be a major regulator of the response. And so what we and others have found is that in geriatric skin, there seems to be a reduced expression of IGF-1 in the dermis of the skin, and IGF-1 seems to supply the epidermis and keratinocytes in the epidermis with that IGF-1. And the level of IGF-1 may impact how the keratinocytes in the epidermis respond to UV. And so what we did was to then do very similar experiments where we took discarded human skin explants ex vivo. What we did then was to treat topically with an IGF-1 receptor inhibitor. So again, we treated topically with either vehicle, DMSO, or 20 micromolar AG538, which is an IGF-1 receptor inhibitor. And we think this will, again, mimic what may happen in geriatric skin. So geriatric skin expresses less IGF-1. And so if we block the IGF-1 receptor with a drug, it's going to mimic that functional effect we would see in geriatric skin. And so then we did very similar experiments. We exposed the skin to between 700 and 800 joules per square meter UVB, incubated for 1 to 2 hours, and then harvested tissue, harvested the epidermis, and then do Western blot. And then we again look for PCNA mono-ubiquitination and total PCNA. And we see very similar response. So in all cases, UV induces an increase in the level of PCNA mono-ubiquitination, but that response seems to be higher when we block IGF-1 signaling. So in skin treated topically with AG538, there seems to be a little bit higher level of this PCNA mono-ubiquitination than in vehicle treated skin. And that looks very similar to what we see in young versus geriatric skin. Again, we think this model of modulating IGF-1 signaling via this IGF-1 receptor inhibitor may mimic what happens in geriatric skin. So this is, again, in figure 2 is essentially looking at, again, normal geriatric versus young adult skin. Figure 3 is modulating IGF-1 signaling in skin ex vivo. We next wanted to move into a more in vitro approach to better understand this. We took a telomerase immortalized keratinocytes. So these are neonatal foreskin keratinocytes that were more immortalized with the telomerase. And then what we did was to treat these cells, cell culture media, treat these cells with either vehicle DMSO, AG538, again, to block IGF-1 signaling, to mimic what we think will happen in geriatric skin, then expose the cells to UVB. And we can see in both cases here, we do see an increase in PCNA mono-ubiquitination relative to PCNA, as shown here. But we see a little bit higher level. So you can see that this span here is a little bit more intense than in the vehicle-treated skin. So we see, again, a little bit higher, stronger response when IGF-1 signaling is inhibited than in vehicle-treated cells. Again, these were telomerase memorialized keratinocytes. We wanted to repeat this in another cell lines. We used Hackett keratinocytes, and again, another model often used to study keratinocyte biology and responses to UV. And we see very similar phenotyping, and we see a stronger response. We see higher levels of this mono-ubiquitinated PCNA in cells in which the IGF-1 signaling system has been blocked. So again, these experiments A through C are all using an IGF-1 receptor inhibitor. And that is quite convenient, but it may not be the best way to understand this process. So we also wanted to confirm this with another approach where we can modulate IGF-1 directly. And so these enterokeratinocytes, we grow them in a very specialized medium where we can remove or include IGF-1 in these cell culture media. And so that's what we did. We plated the cells, grew them for a couple of days, then deprived the cells for 24 hours of IGF-1. So again, no IGF-1 in the media or the media contained IGF-1. Then we exposed to UV just like in figure 4A, and then did Western blotting for PCNA mono-ubiquitination. So we can see, again, a very similar response. We see a stronger response, higher levels of this PCNA mono-ubiquitination when IGF-1 is depleted, which will again mimic what happens if you were to directly block the IGF-1 receptor. So next, I want to better understand sort of the functional consequences of that. So again, as I said in the introduction, the purpose of this PCNA mono-ubiquitination is thought to be to recruit specialized DNA polymerases. And there are a variety of polymerases that potentially can be used to replicate UV photoproducts. The major one that's thought to do it in an accurate way is POL-eta. So POL-eta is actually mutated in a variant form of xerodermal pigmentosum. And it has, as many other TLS polymerases, but they have a PCNA, it has a PCNA, a ubiquitin binding domain. So the idea is it can help the recruitment of the polymerase to the site of DNA damage is facilitated by the mono-ubiquitination PCNA coupled with that a domain in the protein that binds ubiquitin. So what we did was to treat cells with either vehicle or the IGF-1 receptor inhibitor, exposed to UVB, and then after a period of time, I believe this is sure I have in the detail maybe in the method section, probably a half an hour or 1 hour after UV, we harvested the cells, and what we did was to lyse them to enrich for chromatin. So in this case, we're looking at cells that are within the cells are the proteins found in the chromatin or on the chromatin associated with DNA, which is sort of mimicking what happens, represents what's happening to the proteins actually at the site of damage in a global sense. Or we could look at proteins sort of soluble remaining in the rest of the cells. And so if we look at the chromatin fraction, we can see in the vehicle-treated cells, we see a nice increase in the level of POL-eta on chromatin. So it looks like UV is inducing a POL-eta recruitment to chromatin. This is, again, a representative experiment. And B is a Western blot, or an average of 3 different experiments, Western blots showing about a 2.5-fold increase. But when we block IGF-1 signaling with the AG538, we don't see this recruitment. And so we see essentially no change in POL-eta levels in chromatin. Now, if POL-eta is not being recruited, potentially there are other TLS polymerases that may be recruited. So POL-iota and REV1 have both been implicated in mutagenesis associated with UV exposure. And so what we did was to probe these chromatin lysates for those proteins. And in the absence with the IGF-1 normally there, again, we certainly see some level of basal protein, POL-iota and REV1 on the chromatin. After UV, it doesn't seem to change a whole lot. So in figure C, we've quantified that. It doesn't really change much after UV. But when IGF-1 signaling is blocked, now we start to see, again, it's a very modest effect, but it's reproducible, an increase in POL-iota and REV1 on chromatin. So you can see it goes up, maybe 20 to 35% or so. So it's a modest effect. And maybe there's multiple TLS polymerases that are recruited when POL-eta is not recruited to sites of DNA damage. So we think this may contribute to detrimental effects of UV radiation, potentially mutagenic effects. And we'll see this in a minute. So again, these experiments in A, B, and C are all using an IGF-1 receptor inhibitor. We also repeated this experiment in cells where we, just like in figure 4, where we grew cells in the presence or absence of IGF-1, and then did the same sort of experiment. We looked at POL-eta recruitment to chromatin. So in cells with IGF-1 present, again, which would mimic potentially a normal young adult skin, these cells, in vitro at least, we can see POL-eta enriched on chromatin. by nearly twofold after UV exposure. But when cells are deprived of IGF-1, now we don't see this recruitment. So it's essentially flat. It does not increase after UV exposure. And so this was with the entered keratinocytes, and then we see very similar results with Hackett keratinocytes. So again, very similar results using both two different cell lines and both a pharmacological inhibitor of the IGF-1 receptor and also a direct deprivation of the hormone itself from the cell culture media. And so what would be the functional consequences of that? Again, some of these TLS polymerases are mutagenic depending on they get recruited and depending on the type of damage. So what we did was to take Hackett keratinocytes, which are useful for these sort of mutagenesis experiments, and perform something known as an HPRT assay. So normal cells, if they're treated with a drug called 6-thioguanine, they can metabolize the drug. The nucleotide can become incorporated into DNA, and that can essentially prevent cell death or cell proliferation and cause cell death. But if there is a mutation at the hypoxanthine phosphorybtransferase gene, or the HPRT gene, now the cells are unable to metabolize that drug and they become resistant to it. So with very low frequency, we can see that after DNA damage exposure, or exposure to things that generate DNA damage that can cause mutations, we see an increase of these numbers of mutants. And so what we did here was to treat cells with either vehicle, DMSO, or AG538, and expose them to UVB, 50 joules per square meter. And then 5 hours later, we changed the media to get rid of the drugs. And then 5 days later, we actually replated the cells with 6-thioguanine to detect cells with mutations at the HPRT locus. So in the vehicle-treated cells, we see about, again, it's a very low number, about a total of 10 mutants per million cells plated. So again, it's a very low mutation frequency. But in cells where we block the IGF-1 signaling with the AG538, we see about a 3.5-fold or so increase in the number of mutants. Again, consistent with the idea that IGF-1 signaling is somehow preventing mutagenesis. And this may be due to the recruitment of other polymerases. So again, pol-eta can accurately replicate across UV photoproducts, but if it's not present, other polymerases will have to be recruited to allow continued proliferation in the presence of unrepaired UV photoproducts. And so we think that these other polymerases might contribute to that increased mutagenesis in these cells. So that's, again, a brief summary of what we did in this work. Again, really the take-home message is that for the first time, we can actually detect this mono-ubiquitination PCNA in human skin. This is actually the first report, I believe, of any showing it in any sort of tissue, mouse, human, or whatnot. And then we also provide some evidence that the actual functional consequences of that decreased mono-ubiquitination in terms of decreased recruitment of pol-eta, which normally replicates UV photoproducts, that is altered by altered IGF-1 signaling, and that other TLS polymerases might be recruited instead, and ultimately this leads to increased mutagenesis.